Hey, I'm Solomon. Welcome to the channel. So, in my last video, I covered the Holocene out of Africa migrations, which was a major movement of Africans deep from within sub-Saharan Africa into new regions of Europe, the Middle East, and Asia. And in my last video, I got this really great comment from Valentine Akolo. Shout out to him. And he makes a really good point. That being that the region we now call the Middle East was at one time referred to as Northeast Africa, and that the term Middle East is really a modern colonial fabrication created after the construction of the Suez Canal. These regions used to be referred to as Northeast Africa. In fact, a lot of the terms we use to refer to places outside of Africa are modern colonial fabrications, like Eurasia or West Asian. We usually refer to people in the Middle East as West Asian, but this kind of alludes to them being more related to people in, say, China or Siberia. But people in the Middle East, in Palestine, in Israel, in Arabia, are 100 times more related to people in Nigeria than they are to people in Siberia. That's why I can go to Nigeria today, and tens of millions of people speak Afro-Asiatic languages. And I can go to Ethiopia, and tens of millions of people speak Afro-Asiatic languages. And I can go to Israel or Palestine, tens of millions of people speak Afro-Asiatic languages. All of these people are related. They all have a shared African language and African ancestry. And I think this is something a lot of Arabs and Jews aren't aware of. But during the age of the Green Sahara, black Africans used to live in those regions, in Israel, in Palestine. A lot of their ancestors look like me and are from where I'm from. So, as Okolo said, this term Middle East is a new one. Historically, and even in some modern archaeological contexts, this region is still considered a part of Africa. Why is that? Because it's physically attached to Africa. There's nothing stopping gene flow or movement of peoples between these two regions. This is especially true during green Sahara periods, which last for thousands and thousands and thousands of years. These are the conditions when humans first left out of Africa. So, of course, the first modern humans to call the Levant home, the first Levantines or Israelites, were of course black Africans. We have the remains of African people living in the Levant over 100,000 years ago. This is a reconstruction of an ancient woman from Kafsa. Her people migrated from Africa into Israel over 100,000 years ago, their burials being some of the oldest on the planet. These early African migrants took with them much of their ritual practices, artistic practices, and technological practices first developed deep inside inner Africa. For example, in Kafsa, we find the second oldest piece of jewelry in the world. These perforated shells were stained with ochre and then strung around the neck with string. This early practice of personal ornamentation was similarly shared in North Africa, where we find much older necklaces of ochre-stained shells. These finds are significantly older, going back over 150,000 years, and are the oldest jewelry ever found in the world, where the use of ochre to adorn and decorate objects on the body goes back over 300,000 years. The oldest workshops found for the manufacture and process of ochre being found in the Blombos caves of South Africa, as well as some of the oldest examples of human artistic expression on the planet. These early Africans maintaining networks of exchange and interaction throughout the African continent, whether that be in South Africa, North Africa, or Israel.
In the early days of our species, different groups must have been connected in some way. Perhaps they met, mated, and shared genes. That's what anthropologist John Hawkes thinks happened. He believes that social networks existed across the continent. It only needed each group to expand as far as the next for contact to be maintained over huge distances.
All over Africa, archaeologists have found shells like this. This is a Nassarius shell, and it has a hole in it because this shell was strung on a string and worn as an ornament. They've been found in North Africa, in South Africa, and in Israel, thousands of miles apart. Same kind of shell, the same technique, the same intent. Anybody can pick up a shell and be interested in it. But thinking of how to drill a hole and wear it and create a signal, that's a really distinctive idea. That kind of long-distance movement of ideas suggests that people are moving into each other's territories. It's not only ideas that are spreading, it's also genes. That's what humans do when they're in contact with each other. They start mating, they're having babies. You start to see populations merging together.
The movement of prehistoric people was affected by the climate, which fluctuated over thousands of years. In bad times, Africa was so dry, the Sahara was an uncrossable barrier. But in good times, when the climate was wet, the desert disappeared. Any adaptation that emerged in one part of Africa could spread to other parts of the continent. Just as easily as shell beads.
Thus, the first people in Israel were the exact same as they were in Africa, with the same ritual practices, cultural practices, and technology. These early African Israelites were also some of the earliest modern humans to bury their dead. As can be seen here in Kafsa, this is a reconstruction of a young boy buried in Kafsa over 120,000 years ago. These early African residents of Israel would eventually disappear from the archaeological record, and it won't be till around 50,000 years ago Africans would once again exit out the Green Sahara, out of Africa, and into Israel. But this time they wouldn't stop there, and would subsequently colonize the entire earth. But this is not the last out of Africa migration, which most people are not aware of. Around 12,000 years ago, there's another Green Sahara period, thus another out of Africa migration. This migration is known as the Holocene out of Africa migrations, or the Mesolithic out of Africa migrations. During the Holocene, around 12,000 years ago, we will have massive waves of Africans from sub-Saharan Africa deep into new regions of Europe and Asia. For example, you'll find that East Africans migrate down the Red Sea hills into North Africa, into the Levant, then into Turkey and Europe. Then you'll find that Western and Central Africans migrate into the Sahara, into North Africa, into Europe, then subsequently into Turkey and the Levant as well. And a large genetic footprint of these ancient Africans can still be seen in these regions till this day. This is a map of haplogroup E, which originates in Eastern Africa. This Y chromosome spread into these new regions during the Holocene out of Africa migrations. It is during this time East Africans also spread the Afrasian or Afro-Asiatic languages into new regions of the Levant and the Fertile Crescent. This is a map of sickle cell anemia. This genetically inherited condition originates in Western Central Africa, in the deep tropical regions of Cameroon and Nigeria. As these West Africans migrated into new regions of North Africa, Europe, and the Levant, they brought with them these new genetic variants. Thus, sickle cell anemia became common amongst the ancient peoples of the Neolithic Revolution, found commonly in ancient pre-dynastic Egyptians, as well as individuals from the Levant, Turkey, and throughout the Mediterranean. So you have this mixture of Western and Eastern and Central Africans throughout the Mediterranean, Turkey, and the Levant, reaching as far as India. Thus, we regularly find the remains of sub-Saharan Africans in all of these regions. To quote Sa et al., the following 40,000 to 50,000 years witnessed little significant population movement within Africa, and variation accumulated regionally, likely due to a fragmented environment. However, the climatic changes introduced during the Holocene, with the peak of the Holocene climate optimum, allowed woodland and savanna adjacent to the Sahara to expand, creating conditions for the Sahel to become a major corridor, both north, south, and east-west. The expansion of L3 lineages in Eastern Africa and L2 lineages in West Africa attest to the rise in human mobility across the Sahel during this period of improved climate. Movements northwards of Eastern, Central, and Western populations also led to the arrival of sub-Saharan mitochondrial DNA lineages in North Africa and eventually Europe, which was most likely the largest movement of people from sub-Saharan Africa into Europe and Asia since the out of Africa migration. Following the out of Africa migration 60 to 70,000 years ago, significant numbers of sub-Saharan lineages did not disperse from the continent again until the early Holocene. At this time, the climate optimum and the consequent expansion of equatorial forests allowed movements that had previously been limited. Populations were able to cross a greener Sahara to North Africa and Southwest Asia, and from there into Europe, as suggested before, demonstrating the continuing role of climatic oscillations in shaping continental genetic structure. The genetic input from sub-Saharan populations into North Africa points to two major sources. One is West Africa, reinforcing the trans-Saharan movements made possible by the more favorable environmental conditions. The second is Eastern Africa, a likely source of pastoralism in this period. And two things are very important to keep in mind when we talk about these ancient migrations. For one, these African migrations are not migrating into heavily populated areas. There is only about 4 million people on the entire face of the earth at the beginning of the Holocene. And two, the Holocene out of Africa migrations are only 25,000 years after the last out of Africa migration. So people look very, very different throughout Eurasia than they do today. For example, most Europeans look like this. These older groups of Europeans will be largely wiped out and replaced by Indo-European invaders from Central Asia at the beginning of the Bronze Age. In general, these Indo-European migrations are largely the reason why people in Europe and the Middle East look so different today than they did 10,000 years ago. In general, it is a lack of knowledge of these three major migrations that leads to a lot of the confusion about ancient peoples and civilizations. But basically, you have what's known as the three migration model, which goes like this. Around 50,000 years ago, the Sahara is green and leads to African people migrating out the African continent and populating the rest of the world, spreading technology, art, and human culture to the rest of the planet. Then, at about 12,000 years ago, the Sahara is green once again, and you have large waves of Africans leaving the continent, spreading technology, art, and culture once again. And then, at about 5,000 years ago, the Sahara once again turns from green oasis to desert. The drying out of the Sahara happens to coincide with massive migrations from the Central Asian steppe. In Europe, these Indo-European speaking invaders were known as the Yamnaya. These light-skinned invaders would usher in a new age of violence in these new regions of Eurasia. This is known as the Indo-European migrations, and of course leads to the genocide of much of the original inhabitants of Europe. These three migrations, two from Africa and one from Central Asia, pretty much defines human history. To understand world history properly, you have to understand these three migrations. One Paleolithic, one Neolithic, and one in the Bronze Age. But for now, let us return our focus to the Neolithic and the Mesolithic out of Africa migrations. Of course, these major migrations of Africans into the Levant are going to be hugely important to world history. The Neolithic Revolution is really going to be sparked by the spread of these Africans into the Levant. It's these populations that are really going to be responsible for agriculture, urbanism, and the start of civilizations. The remains of these ancient Africans can be found in ancient tombs and sites stretching from Israel to Europe to India. And as I said, their genetic footprint is still present in modern populations till this day. This is haplogroup E, which we know is the dominant Y chromosome of Africa. This Y chromosome lineage spreading out in large amounts during the Holocene and the Neolithic out of Africa migrations. This particular Y chromosome has a very special polymorphism. This mutation is known as the YAP-ALU insertion, making this Y chromosome YAP positive, while all other Y chromosomes, except for E sibling clad D, are YAP negative. During the Mesolithic, Africans bearing this YAP positive Y chromosome would spread throughout Europe, the Levant, and Asia. To quote Underhill, although molecular genetic evidence continues to accumulate that is consistent with a recent common African ancestry of modern humans, its ability to illuminate regional histories remains incomplete. A set of unique event polymorphisms associated with the non-recombining portion of the Y chromosome addresses this issue by providing evidence concerning successful migrations originating from Africa, which can be interpreted as subsequent colonizations, differentiations, and migrations overlaid upon previous population ranges. Sub-Saharan African populations today are characterized by the presence of 4 non-recombining Y chromosome groups, of which group 3, haplogroup E, is the most frequent. The lineage that acquired the YAP M145 M203 polymorphisms in Africa divided into 2 subclusters. One is found today in Africa and the Mediterranean, defined by the M40 M96 mutations. The other subcluster is found in Asia and is defined by the M174 mutation. Group 3 E lineages are found at high frequencies in Africa and relatively high frequencies in the Middle East and Southern Europe, with occasional occurrences in Central Asia, Pakistan, and America. The M35 M215 subclad cluster of haplotypes fragments at lineage HT4 described previously. We suggest that a population with this subclad of the African YAP M145 M203 PN2 cluster expanded into the southern and eastern Mediterranean at the end of the Pleistocene. These lineages would have been then introduced from the Middle East into southern Europe, and to a lesser extent northern India and Pakistan by farmers during the Neolithic expansion. And of course, as these Africans migrated into these new regions, they brought with them their distinct African facial and skeletal morphology. So what exactly makes someone look black, look African? While of course there is lots of diversity amongst Africans, there are certain traits found throughout sub-Saharan Africa that are shared amongst all black people throughout the continent, as well as the diaspora. For example, this is me, my sister, and my uncle. This is my pops, my uncle, and my sister. And this is my pops and a couple of my 8 uncles. Of course, we're all black, we're all African Americans. But aside from our skin color, what exactly makes us look black? For example, these are Ethiopians. What exactly makes them look black? These are Nubians from Egypt and Sudan. What exactly makes them look black? These are Nigerians and Cameroonians. What exactly makes them look black? Well, we all have an ancient ancestral African facial morphology that's sometimes referred to as a sub-Saharan, Africoid, or Negroid phenotype. In short, we all look like the original peoples of Africa, the original peoples of the earth. This is the ancestral phenotype of Africans going back over 300,000 years. It can even be seen on pre-modern humans, such as Jebel Irhoud.
This is an African skull from a site known as Jebel Irhoud. It's 300,000 years old. 100,000 years older than Omo 1. It belongs to an archaic human, someone who was not yet fully modern.
It's a very ancient-looking skull when you look at it from the side, but it has, especially considering its old age, a very modern-looking face. And by that I mean it has a small face that's tucked underneath the brain case, yet the brain case shape is very elongated, which is what we find in archaic humans and not in modern humans living today.
With his mix of features, the man from Jebel Irhoud appears to be a forerunner of our species. A modern human in waiting.
The start of a species is always blurry and messy. This is a process that takes thousands and thousands of years, sometimes millions of years. And we can see here that modern humans evolved their faces first and their brain cases later.
And of course, this phenotype is present amongst the early African residents of Israel, such as Kafsa and other early residents of the Fertile Crescent. And of course, it is the phenotype of the oldest modern human remains ever found, Homo 1, from Ethiopia, from 200,000 years ago.
Homo 1 stood as tall as a modern-day African and weighed 160 pounds. He hunted giant hog and antelope. But one day, Homo 1 could hunt no more. There's no evidence his bones were ever moved or buried. For 195,000 years, they lay where he died.
The one thing we know about this site, the one irreducible fact about Homo 1, is that this is the place where Homo 1 died. He would probably be off to the side someplace, surrounded by members of his family, his wife, his children, his best friend, his brother, his sister. Maybe caring for him, trying to make him comfortable.
Over the years, John Shea has been collecting evidence about the way people lived at the Omo Valley.
You always look underneath the acacia bush. That's where all the good stuff hides.
He's impressed by their ingenuity.
It's a stone tool. We can tell this because they have features on them that show where they're struck by a hammer. Nature doesn't make objects like this by itself. Very nice. I mean, these people made everything from little tiny pieces like this to great massive pieces like this and everything else in between. Sometimes you find a lot of tools, that means that's where they made them. But isolated little tools like this, it could be they just dropped them, you know, and said, all right, I know I'm going to be coming back here. I'll just leave these tools. So here along the path, if I ever need a cutting edge, there's one right there. It's like having an ATM machine. You don't have to carry big wads of cash with you around cities because you have ATM machines. But these guys, they don't have to carry big, heavy rocks around with them because the landscape's full of stone tools. Think of these things like the little debit cards of prehistory. These are people like us. They had a sense of humor, they had sadness, they had joy. There's no reason, to assume that they're different from us in their emotions and their intellect. Nothing in their body says it. Nothing in the archaeology says it. The only thing that compels us to think that these people are simpler than us is mythology. This idea that earlier is primitive and primitive is simple. These guys survived by their wits. They thought, they planned, and evidently they're pretty successful because their descendants, us, are still here.
There are several things that distinguish the skulls of black people from Africa from other groups around the world, and these identifying traits are used by scientists and forensic specialists to identify the ethnicities of individuals in certain populations. Perhaps the most identifiable trait amongst Africans is alveolar prognathism. This is a genetically inherited trait where the upper maxilla protrudes past the plane of the face. It usually results in very full lips. For example, it's very strong in this young Nubian boy from Egypt, and is commonly found amongst sub-Saharan Africans, as well as the remains of ancient Egyptians, as of course they are largely descended from populations from the Sudan and the Sahara. This is an X-ray of Pharaoh Thutmose I, who also has this very strong genetically inherited trait. This is an X-ray of Queen Tetisheri, wife of Pharaoh Seqenen-Retau, who himself was a Nubian and also had these strong African features as well. This prevalence of prognathism and African traits has been well known for decades. To quote James E. Harris, generally, the dentition of each New Kingdom pharaoh and queen represents a unique combination of dental characters, such as overbite, overjet, intercentral relationship, and molar relationship, which permits the identification of each mummy from the X-rays of the dentition alone. This observation is not surprising, since the teeth or dentition remains one of the most formidable tools available to the forensic specialist. Dental alveolar prognathism, an inherited trait which is normal for the Nubian people, ancient and modern, may be observed in pharaohs Thutmose I, Thutmose II, Siptah, and Merenptah, and most of the queens of the 21st dynasty. Black people also have a very distinctive skull shape. We can see this very clearly on both the mummies of Seqenenre and Tetisheri, both having an extremely long and narrow crania. This young Nubian lad also perfectly exemplifies this typical sub-Saharan African trait. This long-headed cranial shape is referred to as being dotiocephalic, which means long-headed. This is in stark contrast to most European and Asian populations, whose crania are usually much shorter. This is known as being mesocephalic or brachiocephalic. And of course, while there is several different nose shapes across sub-Saharan Africa, the more softer, rounder shape variation is found commonly throughout the continent. This is because Africans usually have a broader nasal aperture, which is a typical heat adaptation, and usually results in a broader, flatter nose, as well as having a very short nasal spine. This is the little bit of bone that supports the cartilage that separates the nostrils. In other groups, it is very prominent, or of a moderate height, where in Africans it is very small or sometimes non-existent. All of these features are what make black people look like black people. And there's actually several other black African specific markers that help anthropologists and forensic specialists differentiate black people's skulls from other groups, such as shape of eye orbits, Africans usually being more rectangular, or the projection of the lower jaw and forehead shape, or the shape and distance between cheekbones and the shape of the forehead. But in general, the biggest clues to sub-Saharan African ethnicity are those big three main traits: prognathism, a broader nasal aperture, and that long-headed dotiocephalic skull shape. And as I said, as Africans migrated into these new regions of the Mediterranean, the Levant, Turkey, and Asia, during the Neolithic, they brought along their ancestral African or Negroid phenotype. Thus, the remains of black people from sub-Saharan Africa are found commonly amongst the peoples of the Neolithic Revolution. These African populations being a very large part of the communities that built sites like the Tower of Jericho and Göbekli Tepe, and they are found in extremely important Neolithic settlements, such as Karahuik in Anatolia or modern Turkey, or Neo-Nicomedia in Neolithic Greece. As we know, sickle cell anemia originates in Western Central Africa over 22,000 years ago, in the tropical regions of Cameroon near Nigeria. As these Western Central Africans migrated across the Green Sahara and into these new regions of the Mediterranean and the Levant, they of course brought with them this genetically inherited illness. Thus, sickle cell anemia, parodic hyperostosis, and falciparum malaria is found commonly amongst the populations of the ancient Levant, Mediterranean, and Egypt, along with these sub-Saharan African peoples. To quote Angel, during the migrations and population increases and decline in health accompanying the 8th to 6th millennium BC farming revolution, spread of the new mutant falciparum malaria crossed the flow of genes from Africa northward, as indicated by parodic hyperostosis increase, abnormal hemoglobins and anemia, and negroid traits, nose breath, and prognosticism. There are two distinct modes in the biological interaction between populations of early Egypt and those to the south in Nubia, and to the east in the Levant, the Aegean, and Mesopotamia. Flow of genes and flow of diseases. In latest Paleolithic hunting times, somewhat isolated population groups, often smaller than 100 people, might be much affected by gene flow and less affected by disease spread, except where the latter also involved natural selection, that is, affected survival and fertility. A striking observation reconfirmed by each new study of latest Paleolithic hunting groups is the decline in health at the time of the change from a hunting economy to the earliest farming economy. Decrease in body size, decrease in jaw and tooth strength and size, usually increase in dental disease, no increase in adult longevity, no improvement in juvenile mortality, and in people settled near marshes, the sudden appearance of parodic hyperostosis, anemia, at fairly high frequency. Against this background of disease, movement, and pedomorphic reduction of body size, one can identify negroid traits of nose and prognosticism appearing in Natufian latest hunters and in Anatolian and Macedonian first farmers, probably from Nubia, via the unknown predecessors of the Badarians and Tazians, and traveling in the direction of sickle cell anemia and thalassemia, and hence also falciparum malaria. The history of this new mutant is extremely important. Many malariologists have assumed that it originated recently in Africa, usually meaning during the Pliocene or Pleistocene. The 2 or 3 millennia of transition from hunting to farming would be time enough for the rise in frequency of abnormal hemoglobins. The mutations are apparently old ones, since parallels occur in the great apes, and for other microevolutionary changes, signs of skeletal anemia occur in both half the adults at Neonicomedea and at Chateauhouic, but are rare or absent in latest Paleolithic North Africans and Nubians, and occur in Egypt in pre-dynastic times. And it should be understood, these sub-Saharan African populations were not completely wiped out with the beginning of the Bronze Age and the Indo-European migration. The remains of these ancient sub-Saharan Africans being found deep into antiquity in regions such as Anatolia or modern Turkey, in areas such as Sagalosas. To quote Rakau, from the Mesolithic to the early Neolithic period, different lines of evidence support an out-of-Africa Mesolithic migration to the Levant by northeastern African groups that had biological affinities with sub-Saharan populations. From a genetic point of view, several recent genetic studies have shown that sub-Saharan genetic lineages affiliated with Y chromosome P and 2 clade have spread through Egypt into the Near East and Mediterranean area, and for some lineages as far north as Turkey, probably during several dispersal episodes since the Mesolithic. This finding is in agreement with morphological data that suggests that populations with sub-Saharan morphological elements were present in northeastern Africa from the Paleolithic to at least the early Holocene, and diffused northward to the Levant and Anatolia beginning in the Mesolithic. Indeed, the rare and incomplete Paleolithic to early Neolithic skeletal specimens found in Egypt, such as the 33,000-year-old Nazlet Cotter specimen, the Wadi Kubania skeleton from the late Paleolithic site in the Upper Nile Valley, the Koranian, Fayum early Neolithic crania, and the naphtha specimen from the Neolithic Naphtha Playa site in the western desert of Egypt show with regard to the great African biological diversity similarities with some of the sub-Saharan Middle Paleolithic and modern sub-Saharan specimens. This affinity pattern between ancient Egyptians and sub-Saharans has also been noticed by several other investigators and has been recently reinforced by the study of Brace et al., which clearly shows that the cranial morphology of prehistoric and recent Northeast African populations is linked to sub-Saharan populations, Niger-Congo populations. These results support the hypothesis that some of the Paleolithic early Holocene populations from Northeast Africa were probably descendants of sub-Saharan ancestral populations. A late Pleistocene, early Holocene northward migration from Africa to the Levant and to Anatolia of these populations has been hypothesized from skeletal data and from archaeological data as indicated by the probable Nile Valley origin of the Mesolithic Mushabi culture found in the Levant. This migration finds some support in the presence in Mediterranean populations, Sicily, Greece, southern Turkey, of the Benin sickle cell haplotype. This haplotype originated in West Africa and is probably associated with the spread of malaria to southern Europe through an eastern Mediterranean route, following the expansion of both human and mosquito populations brought about by the advent of the Neolithic transition. This northward migration of northeastern African populations carrying sub-Saharan biological elements is concordant with the morphological homogeneity of the Natufian populations, which present morphological affinity with sub-Saharan populations. In addition, the Neolithic Revolution was assumed to arise in the late Pleistocene Natufians and subsequently spread into Anatolia and Europe. And the first Anatolian farmers, Neolithic to Bronze Age Mediterraneans, and to some degree other Neolithic Bronze Age Europeans, show morphological affinities with the Natufians and indirectly with sub-Saharan populations, in concordance with a process of demic diffusion accompanying the extension of the Neolithic Revolution. Consequently, one may hypothesize as the most parsimonious explanation that sub-Saharan biological elements were introduced into Anatolian populations after the Neolithic spread and have been preserved since this time, at least until the 11th and 13th century AD, in the population living in the Sagalos territory of southwestern Anatolia. This scenario implies that the affinity between Sagalos and the two sub-Saharan populations, Gabon from West Africa and Somalia from East Africa, is more likely due to the sharing of a common ancestor, and that the major changes and increasing interactions in the Eastern Mediterranean beginning in the Bronze Age did not erase some of the sub-Saharan elements carried by Anatolian populations, as shown by genetic data and the morphological features of our southwestern Anatolian sample. So, as we can see, the Neolithic Revolution was in large part sparked by the movement of large waves of Africans into the Levant and their subsequent spread into Europe and Asia. But these Africans didn't stop just at India. They in fact migrated deeper into East Asia, as well as into the Americas, as we will cover later on. But to understand these Neolithic migrations into the Americas, we first have to understand when the Americas was first populated and by whom. To understand that, we have to go back to Africa, the Levant, and the very beginning of the Paleolithic out of Africa migrations.
This is the Wadi Gadoon, one of the biggest ancient river valleys in all of Arabia, perhaps. It's probably half a mile to the other side. It's dry now, but 100,000 years ago, this thing was full of water. All of Arabia was green, for that matter. It was covered in savannas and lakes, and you have this vast network of river valleys that run through this plateau.
He began to explore the hilltops above the ancient riverbed. And here, he found a field of flaked rocks.
So, this is the stuff I've been looking for all those years along the coast. This is called chert or flint, and it's the raw material that was essential to make the stone tools throughout prehistory. And a site like this is literally covered in a carpet of this material. Prehistoric tool makers would come to a site like this. They would bang away, make their stone tools, leave the refuse behind, and then go off down into the valleys where the water was. So this is a prehistoric workshop.
It was unmistakable. People had passed through here and hung around long enough to make thousands of tools. But who and when? The stones told him all he needed to know.
That is gorgeous. That is a classic Nubian point. This is the tool that was made by the first people that left Africa.
Nubian points came from a part of Africa now in modern-day Egypt. They were made about 100,000 years ago. If the same tools can be found in Oman, it suggests people were on the Arabian Peninsula at the same time. 100,000 years ago.
We're not looking at something that looks like it may have a similarity to an African technology. We're looking at the identical African technology.
If Africans were in Arabia 100,000 years ago, then people were migrating into Asia much earlier than archaeologists thought. 40,000 years earlier. But what drove them on into the unknown? Perhaps it was the climate. Today, northern Africa and Arabia are hot and dry. But 100,000 years ago, this region was very different. The deserts bloomed. The landscape was green. The world was in a wet phase.
The climate here is cyclical. So you have this monsoon system that's circulating through the Indian Ocean, and then every once in a while, it intensifies, it strengthens, and it pushes into the interior of Arabia. Rivers like this that were once dry were flowing again, and so it was almost like the highways were open, and they just filled them up.
It wasn't the beaches that lured people into Arabia, but the rivers, filled by monsoon waters.
What we realized was that people weren't interested in the coastline. They weren't these fishermen who had adapted a new technology and were moving along this new ecosystem. They were the same people as they were in Africa, and they were opportunistic. These river valleys would have been paradise because not only is it the exact landscape that they already knew, it was also unoccupied, so there was nobody to compete with. Some of the qualities that we can see in these people, which we really haven't seen before in prehistory, is they're curious. There was no overpopulation, so they weren't forced out of Africa, but they would wake up every day on one side of the Red Sea and they'd look across, and at some point, somebody couldn't stand it anymore and had to go scratch that itch. I think it was this curiosity that was driving them, and I think that's what we are. That's the defining aspect of our species, is we want to know what's on the other side.
For the first peoples, the way into Asia was open. From Arabia, they migrated eastward into Central Asia, northern India, and beyond. They would have followed a network of rivers, natural corridors through the continent. Southeast Asia, 63,000 years ago. A band of hunter-gatherers on the lookout for food. They're newcomers here, migrants from Africa, at the edge of the known world. People in the tropics tend not to live in caves. There are too many poisonous predators. They use them as a shelter to escape the rain. But for one young woman, this cave will be her final resting place. Her people left Africa and migrated 4,000 miles eastward, following game, looking for new sources of food.
South Asia, Australia, and Melanesia have the special distinction of being the earliest locations on Earth settled by modern humans leaving Africa. Some of these groups of early Africans arriving in these regions as early as 100,000 years ago. These early Africans would have arrived in a very different looking South Asia and Austro-Melanesia. During the Paleolithic out-of-Africa migrations, instead of these regions consisting of several different islands, they were in fact two large conjoined land masses, known respectively as Sundaland and Sahul. Sundaland was connected to mainland Southeast Asia and contained modern countries like Bali, Borneo, Java, and Indonesia, where Sahul consisted of Australia and what is now the islands of Papua New Guinea. Even the islands of the Philippines were one landmass connected to Borneo. These early Africans that would make Sundaland and Sahul their new homes would go on to have one of the oldest continuous population histories outside of sub-Saharan Africa. And one of the things that is immediately noticeable about many of these modern populations in these areas is many of them still look black. Many of these populations, unlike many other regions around the world, have retained their ancestral sub-Saharan African phenotype. Outside of Africa, they're the only populations to retain this skeletal morphology. And it's not just their black skin color, but their actual cranial morphology. Remember, these traits are genetically inherited. Thus, they still have many of the traits of their African ancestors, such as pronounced amounts of alveolar prognathism, that wider nasal aperture, and that distinctive African long-headed dactylocephalic skull shape. There are several different major groups that make up these regions. One is the Negritos, a broad term that encompasses many of the native short-statured black peoples that are descended from the original African inhabitants of Sundaland. Some of these groups include the Onge, or the Andaman Islanders, the Aeta, and the Batak. These dark-skinned groups are often referred to as Orang Asli, or original peoples, by the lighter-skinned Mongoloid peoples that migrated into these regions during the late Neolithic. These groups also include indigenous Australians, referred to in modern day as Australian Aborigines, as well as the various groups of Papua New Guinea, such as the people of Bougainville, or the native Melanesian peoples of the Solomon Islands. These people are the descendants of the original African inhabitants of Sundaland and Sahul over 80,000 years ago. It's often been said, quite inaccurately, that the appearance and skin color of these South Asians and Australo-Melanesians is a coincidence, a fluke. They in fact look black on accident, a process of convergent evolution. This is a complete lie. These people have this skin color and this phenotype because of their ancestors. It is the skin color and phenotype of their ancestors from Africa. These groups arrived there with that skin color and that phenotype and have simply retained it over the millennia. We in fact know this because these people share several genetic variants, or SNPs, that are identical by descent with Africans, meaning that several portions of their DNA are the exact same as their African ancestors and their relatives in modern Africa. But before we touch on that, what exactly is an SNP? And what exactly makes groups of people related? Well, the first thing that should be understood is that all human beings, no matter where they're from, are 99.9% genetically identical. All human beings are from Africa and have black African ancestors. These were fully modern humans with speech and art and invention and technology that left the African continent and populated other regions of Afro-Eurasia. These were not hominids or pre-humans, but modern black human beings. This is why all human beings have nearly identical DNA, being 99.9% the same. That 0.1% accounts for all the differences we see amongst modern groups of people. These differences are caused by mutations that are known as SNPs or SNPs. These SNPs, or variations in DNA, account for differences like eye color or skin color or height or hair texture or certain diseases. Thus, we can say certain groups are related by their shared SNPs or genetic variants, as groups with certain mutations have a common set of ancestors. So, what causes these SNPs or SNPs? And what does it stand for? SNP stands for single nucleotide polymorphism. Single, of course, means one, and nucleotides are the building blocks of your DNA. And polymorphism simply means change or mutation. To start off, your DNA is made up of 4 nucleotides, known as adenine, cytosine, guanine, and thymine. These nucleotides attach to each other to forge chemical bonds known as base pairs, which connect 2 DNA strands. These base pairs are labeled as A, C, G, and T. And these base pairs serve as the instructions for coding proteins to make up your body. As a cell copies its DNA before dividing, it occasionally makes typos. So sometimes a single base pair will be substituted, deleted, or duplicated. And these changes or mutations in base pairs can have no effects or major effects. And these mutations, or single nucleotide polymorphisms, account for the major changes we see amongst human populations. These mutations, or SNPs, are then subsequently passed down to subsequent generations. Thus, we can trace the origin of populations by their shared polymorphisms, or SNPs. For example, let's take a look at the Y chromosome. All Y chromosomes originate in Western Central Africa. It's in these regions we find Y chromosomal Adam, the ancestor or father of all men and paternal lines that walk the earth. It's here in West Africa we find A00, the beginning of all paternal lines of Africa and of all paternal lines of the human race. To quote Fernando L. et al., we report the discovery of an African American Y chromosome that carries the ancestral state of all SNPs that define the basal portion of the Y chromosome phylogenetic tree. We sequenced 240 kb of this chromosome to identify private derived mutations on this lineage, which we named A00. We then estimated the time for the most recent common ancestor of the Y tree as 338,000 years ago. This is the beginning of the Y phylogenetic tree. All Y chromosomes are versions of this original Y chromosome. The only difference is they've acquired SNPs or mutations that allow us to tell them apart from the original Y chromosome. So a BY chromosome is simply an AY chromosome with additional SNPs or mutations that let us distinguish it from that original AY chromosome. Same thing with a CTY lineage. It's a version of a BT lineage with additional SNPs, namely mutation M168 and M294. Thus, all letters of the Y phylogenetic tree are simply a version of the preceding Y chromosome, just with additional SNPs or mutations. So, while we typically label the Y phylogenetic tree A through R, if we wanted, we could label it A0 through A17, because in fact, all Y chromosomes are a version of that original A with simple additional SNPs or single nucleotide polymorphisms. The first 6 mutations of the Y chromosome tree occur in Africa, namely A, B, C, D, E, and F. All of these Y chromosomes have an African origin. Then Africans carrying one branch of haplogroup D and haplogroup CF will leave out the African continent and subsequently populate the rest of the world. And it's important to know the people that left Africa were still very much African, and they very much looked African for thousands and thousands of years. And in some cases, they still do look African. I think people have this idea in their head that once people left Africa, they stopped being related to black people and people in Africa, which is not the case at all. As people migrated into new regions, they simply accumulated different mutations. But I can't state this enough. The mutations that happen inside of Africa are no different than the mutations that happen outside of Africa. They're all simple mutations on the exact same original African genome. So, as I said, we can see how certain populations are related by their shared SNPs, or SNPs. For example, sickle cell anemia is caused by a very specific SNP. This switch, or nucleotide polymorphism, causes a mutation in the protein that makes the red blood cells, causing many of the patient's blood cells to take a rigid sickle shape. And of course, these alleles, or SNPs, spread out in large numbers during the age of the Green Sahara. Thus, all peoples with sickle cell, whether they live in Africa or Eurasia, have a shared West African ancestry. So, while a mutation like sickle cell causes changes we can see on the inside of the body, some of these mutations, or SNPs, cause changes we can see on the outside of the body, such as skin color. So, for example, the SNP that causes light skin is only about 20,000 years old, and that mutation won't become widespread into regions like Europe till the Indo-European migration, around 5,000 years ago. Whereas the mutation for light skin in East Asians is a little different, being only about 6 to 12,000 years old. So both of the typical light skin phenotypes we usually associate with both Europeans and East Asians are very new and caused by very recent mutations or polymorphisms and subsequent migrations of peoples from colder regions. For example, the mutation that causes depigmentation in East Asians is located on a completely different part of the genome than Europeans, namely the OCA2 gene on the 15th chromosome. Remember, SNPs are simply mutations in base pairs, made up of adenine, cytosine, guanine, and thymine. These base pairs are read off in triplets known as codons, and of course changes in these instructions, in these base pairs of A, C, G, and T, and how they're read, causes changes that we can sometimes visibly see in people. In this case, it causes changes in skin color. To quote Murray, two OCA2 polymorphisms, RS1800414 and RS746533330, have been associated with pigmentation in East Asians. We explored the distribution of these markers in a panel of samples from populations around the world. The derived allele of RS1800414 has high frequencies in a broad East Asian region, whereas the derived allele of RS746533330 is primarily restricted to northern East Asia. The haplotype analysis suggests that the haplotypes carrying the derived alleles for each polymorphism arose independently from the same ancestral haplotype, using the markers RS7170451 and so on to define the haplotype block. Our results indicate that from the ancestral haplotype AAGAGCAGGTT, or adenine, adenine, guanine, adenine, guanine, cytosine, adenine, guanine, guanine, thymine, thymine, a non-synonymous mutation at RS1800414 originated the haplotype AGGAGCAGGTT, or adenine, guanine, guanine, adenine, guanine, cytosine, adenine, guanine, guanine, thymine, thymine. And another non-synonymous mutation independently originated the haplotype AAGAGCAAGTT, or adenine, adenine, guanine, adenine, guanine, cytosine, adenine, adenine, guanine, thymine, thymine. Both derived haplotypes then increased in frequency in different regions of East Asia. The haplotype AGGAGCAGGTT is now the most common haplotype in a broad region of East Asia, whereas the haplotype AAGAGCAAGTT has become the most prevalent in northern East Asia. Several lines of evidence indicate that this increase in frequency may have been the result of positive selection favoring light skin in high latitude regions. Both the derived alleles are non-synonymous variants predicted to have a functional effect, and both have been associated with lighter skin pigmentation in East Asian populations. In addition, several studies have identified signatures of positive selection in the OCA2 region in genome-wide scans in East Asian populations. The geographic distribution of the variant strongly suggests that these two mutations arose after the separation of European and East Asian populations. This is supported by a recent study that dated the derived G allele of the OCA2 RS1800414 polymorphism to 10,000 years ago. To our knowledge, there has been no attempt to date the polymorphism RS7465330. We use the dense genome-wide SMP data available for the HGDP CEPH panel to estimate the ages of the derived alleles at RS1800414 and RS7465330 in East Asian populations. We used a method that relies on the decay of haplotype sharing of the ancestral genomic segments on which the derived mutations occurred. Under these conditions, and assuming a generation time of 29 years, we estimated the age of the derived allele at RS7465330 to be 6,835 years old, with a 95% confidence interval between 1,070 and 12,798 years ago. The estimated age of the derived allele at RS1800414 is quite similar, at 6,397,000 years ago, with a 95% confidence interval of 1,183,000 years ago and 11,446,000 years ago. Recent ancient DNA studies, which have characterized dense genomic data in Eurasian individuals spanning a broad archaeological period, e.g. from hunter-gatherers to individuals living in the Bronze Age, have provided important information about the temporal distribution of genetic markers associated with pigmentation variation in Europe, and have strengthened the case for selection operating in the pigmentation-related genes in this region. Similar studies in East Asia have the potential to clarify the major events that have shaped the interesting distribution of the two non-synonymous variants of the OCA2 gene in this vast area. In this respect, it will be important to consider not only potential selective effects, but also the major population movements that have taken place in this region during the past 15,000 years. And this brings us back to the peoples of South Asia and Australo-Melanesia. As I said earlier, these people are unique amongst populations living outside of Africa, as they've retained their ancestral African phenotype and skin color. And as I said, this is not due to coincidence or convergent evolution, but their shared African ancestry with their relatives in the African continent, having several shared SNPs or genetic variants that are identical by descent with Africans, namely mutations on several genes such as DDB1 on the 11th chromosome, OCA2 on the 15th chromosome, and TMIM138 on the 19th chromosome. These are only a few of the shared SNPs between Australo-Melanesians and African populations. These specific mutations only being shared by these two populations. To put it in layman's terms, they look African because they're from Africa, even though they don't reside there currently. For example, this is me as a little kid, and my sister, and my dad, and my cousin. And these are Papuans, and Aborigines, and Melanesians. Why do we all look alike? Why do we all look like family? Why do we all look like we're related? That's because we all have the same genetic variants. We all have the exact same ancestral African SNPs dictating our phenotype. To quote Crawford, individuals from South Asia and Australo-Melanesia share variants associated with dark pigmentation at MFSD12, DDB1, TMIM138, OCA2, and HERC2 that are identical by descent from Africans. This raises the possibility that other phenotypes shared between Africans and some South Asian and Australo-Melanesian populations may also be due to genetic variants identical by descent from African populations, rather than convergent evolution. This observation is consistent with a proposed southern migration route out of Africa 80,000 years ago. Alternatively, it is possible that light and dark pigmentation alleles segregated in a single African source population, and that alleles associated with dark pigmentation were maintained outside of Africa only in the South Asian and Australo-Melanesian populations due to selection. So, as we can see, the appearance of these populations is not from coincidence, but their actual African ancestry and shared African genetics. So, now that we know that, when exactly did people first arrive in Sundaland and Sahul? When did the African ancestors of the Australo-Melanesians first arrive? Well, the three groups that left out of Africa carried three distinct Y chromosomes, as we know, D, C, and F. The DY chromosome has its origin in Western Africa. There is one place in Africa you can find the D haplogroup, and that is here, its place of origin, West Africa, Nigeria. To quote Haber et al., here we reinvestigate a rare deep-rooting African Y chromosomal lineage by sequencing the whole genomes of three Nigerian men described in 2003 as carrying haplogroup DEY chromosomes, and analyzing them in the context of a calibrated worldwide Y chromosomal phylogeny. We confirm that these three chromosomes do represent a deep-rooting DE lineage, branching close to the DE bifurcation, but place them on the D branch as an outgroup to all other known D chromosomes, and designate the new lineage D0. The most favored model involves an origin of the DE lineage within Africa, and D0 and E remaining there, and migration out of the three lineages C, D, and FT that now form the vast majority of non-African Y chromosomes. The exit took place between 50,300 and 81,000 years ago, and most likely between 50,300 and 59,400 years ago. This work resolves a long-running debate about Y chromosomal out of Africa back to Africa migrations, and provides insight into the out of Africa expansion more generally. D is the dominant Y chromosome of the Onge and the Andaman Islanders, indicating their ancestors had a Western Central African origin. These early West African migrants also were the first people to populate regions like Japan. These early West African populations, bearing that DY chromosome, also were the first to populate the regions of Tibet, where the DY chromosome still dominates till this day. To quote Mandal, the Andamanese stand out in our dataset because of their haplogroup D, which in India has been reported only in the Andaman Islands and is present in all of the 5 individuals we sequenced. This haplogroup is especially interesting as it has a deep genealogy, sharing a 4,000-year more recent ancestry with the African haplogroup E than all other haplogroups found in the out of Africa populations. This relationship with African populations has been a basis for postulating a distinct early out of Africa migration, differentiated from the later and more widespread Asian expansion. This proposal is in stark contrast to findings from the autosomal data, where the Andamanese showed a common ancestry with other Asian populations without a trace of contribution from a putative early out of Africa population. In addition, this haplogroup has been found at high frequency in Japan and in Tibet. We found that this haplogroup in the Andamanese has a deep substructure, which is a much higher ratio than for other Indian populations. This suggests that the Andamanese Y chromosomes have had a long-lasting separation from the other populations examined, and that male gene flow into the Andaman Islands has been limited. As this haplogroup is found at high frequencies in Japanese individuals from Tokyo, from the 1000 Genomes Project, it is possible to analyze the divergence times between the two populations for both the haplogroup D Y chromosomes and the autosomes. The divergence time between Andamanese and Japanese haplogroup D Y chromosomes is 53,000 years ago. while the divergence time between haplogroup D and O chromosomes is 77.3 thousand years ago. In contrast, the autosomal divergence time is 52,000 years between Andamanese and East Asians. This possible discrepancy between Y chromosomes, divergence between D and O haplogroups, and autosomal divergence between Andamanese and East Asians could potentially be explained by two different hypotheses. It could be caused by two different settlements. People with haplogroup D first populated Asia, including the more external localities like the Andaman Islands and Japan. Later in Japan, this haplogroup was partially replaced by haplogroup O individuals. According to this model, haplogroup D is more frequent in the two extremes of the archipelago as a consequence of this later replacement. Or B, if a single main out of Africa event happened, haplogroup D, along with other haplogroups in the other out of Africa populations, i.e., C and macrohaplogroup F, were already present in the initial out of Africa population, but later haplogroup D was lost from other populations by random drift, except in the Andaman Islands, Japan, and Tibet. If the first hypothesis were true, even if the genomes of Japanese O and Japanese D had homogenized, we would expect the Andamanese and Japanese D to share more derived autosomal alleles with each other than with other East Asian populations. We found that this is not the case. In fact, as seen from the autosomes, the Andamanese are an outgroup to East Asian populations, diverging in a trichotomy with East Asians and Indians. Another important Y chromosome of these early out of Africa populations is haplogroup C. This is the dominant Y chromosome of the indigenous Australian Aborigines and also has its origin in Africa as well. To quote Halast, here we show that phylogenetic analysis of haplogroup C, D, and FT sequences, including very rare deep-rooting lineages, together with phylogeographic analysis of ancient and present-day non-African Y chromosomes, all point to East-Southeast Asia as the origin 50,000 to 55,000 years ago of all known surviving non-African male lineages, apart from recent migrants. There is one region of the genome with the potential to inform about these events in a unique way, the Y chromosome. This is because its male-specific portion provides haplotypes from which a detailed, calibrated phylogenetic tree can be created. Several such trees have been constructed independently and are all consistent in being dominated by a massive expansion of non-African Y lineages during the key interval of 50,000 to 60,000 years ago, starting from a single haplogroup designated CT. Taking into account a rare African D0 lineage and the time frame summarized above, we have argued that the initial splits within CT are likely to have occurred inside of Africa before the exit, and that three lineages, C, D, and FT, were carried out by the ancestors of present-day non-Africans. Each of these three lineages subsequently expanded, C and D moderately and FT massively. It's important to remember that during the Paleolithic, both Australia and Papua New Guinea are one landmass known as Sahul. These populations are some of the oldest on the planet, and in fact, modern Papua New Guineans still have up to 2% of an even earlier wave of human migration from Africa, dating back over 100,000 years ago. To quote Pagani, we find a genetic signature in present-day Papuans that suggests that at least 2% of their genome originates from an early and largely extinct expansion of anatomically modern humans out of Africa. Together with evidence from the Western Asian fossil record, an admixture between anatomically modern humans and Neanderthals predating the main Eurasian expansion, our results contribute to the mounting evidence for the presence of anatomically modern humans out of Africa earlier than 75,000 years ago. The paths taken by anatomically modern humans out of Africa have been the subject of considerable debate over the past 2 decades. Fossil and archaeological evidence and craniometric studies of African and Asian populations demonstrate that Homo sapiens were present outside of Africa 120,000 to 70,000 years ago. However, this colonization has been viewed as a failed expansion out of Africa. since genetic analysis of living populations have been consistent with a single out-of-Africa migration followed by serial founder events. In conclusion, our results suggest that while the genomes of modern Papuans derive primarily from the main expansion of modern humans out of Africa, we estimate that at least 2% of their genome sequence reflects an earlier otherwise extinct dispersal. The inferred date of the early out-of-Africa split time is 120,000 years ago and is consistent with fossil and archaeological evidence for an early expansion of Homo sapiens from Africa. These early African migrants in South Asia would be the first modern humans to make a significant sea voyage, becoming the first peoples of Sahul, bringing with them their culture, technology, and religious practices from their native homeland of Africa.
Southeastern Australia, 42,000 years ago. A man is buried by a lakeside. He's a clan chief or something similar. Important enough to be buried with ceremony. He's known to archaeologists as Mungo Man. Here, at the very ends of the earth, is some of the earliest evidence of our yearning for spirituality. Mungo Man was discovered in 1974 by geologist Jim Bowler.
This is an amazing view of the landscape. It's wonderful to be back here.
One summer's day in 1974, Bowler was riding his bike at Lake Mungo when he saw something unusual eroding out of the dune.
I spotted a tip of white bone exposed on the surface of the sand, and it was over near here, just the top of cranium coming out of the sand. And when I brushed away the lower part, the jaw was there. So immediately I recognized I was standing on the burial of a human remains.
The bones were 42,000 years old, twice as old as remains found anywhere else in Australia. What was most striking was they'd been stained with a natural pigment, ochre.
There's no ochre within 100 kilometres or more from here. The ochre had to be brought in. It had to be prepared. All that preparation for the moment of burial, that went on for days.
Humans had already been using ochre for thousands of years in Africa as a way to decorate their bodies or treat their hides. But here, it was an integral part of an elaborate ritual ceremony.
He was laid out as the way we would lay out my own father in his funeral coffin, and he was then ceremonially farewelled. It was not just the mourning of death. It was even perhaps something of a celebration of life, really.
Mungo Man is not the only body Jim Bowler has found by the lakeside. A quarter of a mile away, he discovered fragments of a female skeleton that became known as Mungo Lady.
This is the sort of horizon in which I found the bones of Mungo Lady.
When anthropologists put together all the fragments from Mungo Lady, they had evidence of another funeral ritual from the same period, 42,000 years ago. Mungo Lady's bones had been burnt, perhaps to make them more brittle. Then they were broken into tiny pieces. Lake Mungo was clearly a home for a community of people with a rich, complex culture. But who were these people living at the edge of the world? Where did they come from? When did they arrive? How did they get here? Lake Mungo is tucked away in the southeast of the country, an isolated corner in the world's most isolated continent. It's a mystery how people ever reached Mungo, because there's so little archaeological evidence from that period anywhere in Australia. But there are clues to be found in DNA. At the start of the 20th century, British anthropologist Alfred Haddon went to Australia to record the customs of the indigenous people and collect samples of their hair. His collection remained in a museum, gathering dust, until a Danish geneticist, Eske Willerslev, got to analyze the samples.
The beauty about hair is that it's not porous like bone and teeth. So it means that all contamination from all people who have handled it over the years is all sitting on the outside of the hair. So we can basically just put it into bleach and then get rid of the contamination, and the DNA remaining is the one from the individual himself. This hair collection from Cambridge has probably many times people have asked themselves, what should we use this hair for? And then 100 years later, I mean, this turns out to be actually a piece of gold for a geneticist.
The hair sample came from a man in the outback of Western Australia, which had barely been visited by Europeans at that time. So the DNA was pure Aborigine. This was the first time anyone had sequenced an Aboriginal genome. From this one sample, it was possible to unlock the ancient history of a people.
Archaeology tells us that humans were present in Australia at least 40,000 years ago. The question is, of course, who were these people? How were they related to present-day Aboriginal Australians or to other peoples in the world, for that matter? And this is, of course, where genetics comes in as a very powerful tool. You can start tracing the ancestry of Aboriginal Australians. You can actually find out when did they diversify from other living people, when did they come out of Africa. We can link biologically these individuals to other human beings in space and time.
Analyzing the DNA was like looking into the deep past, back to the first wave of humans migrating out of Africa. Rather than exploring Asia, the first Australians headed south. They split from the rest of humanity 60 to 70,000 years ago. And then they were on their own.
Aboriginal Australians are descending from the first people moving out of Africa and getting into Australia. They are direct descendants of the first modern human explorers.
These explorers must have reached the southern tip of Asia. Instead of turning back, they adapted to life at the coast. Hugh Barton is an archaeologist and an expert on the lives of coastal hunter-gatherers.
Coastal environments like this with, sort of water, lagoons, sort of back swamps, potentially mud flats, mangroves, they're really good places for food for hunter-gatherers. There's, you got shellfish, fish, clams, eggs, worms and grubs. There's lots of rich, high-energy foods that people can take from these environments. But if you've got a group of, say, 5 to 10 people, you might clean out an area of its sort of high-rank, good quality resources, within days or weeks. So that can be a real trigger point for people deciding then, well, we need now to move on to a new location.
For hunter-gatherers living at the coast, relocating could mean a trip offshore.
Perhaps you can see small islands, not too far away. So people will go out ahead, they'll find out, they'll come back, they'll communicate to the group information about what they found, and they might make then a decision as a group to go on to potentially, and maybe hop across to another island.
Once people started island hopping, Barton thinks it was only a matter of time before they stumbled on a way to reach Australia.
Most of us look at the ocean now, kind of see that as a real barrier, and it's kind of to be avoided. You want to be on the land because that's okay, that's solid, But if you've got a group of people that maybe they've got some simple watercraft, they're out, they're fishing, once they've gone out and they've gone out, who knows, maybe they were brave enough to go out overnight. But then suddenly they start to pick up signs that might tell them that potentially there's a land mass out there. Could be clouds, could be birds, could be driftwood. They might think, actually maybe it's worth kind of pushing on a bit here. Maybe for 20 years, 100 years, they don't really get there. But at some point, someone goes that little bit further, just takes that further risk, whatever the circumstances, gets across. The next thing you know, they're bumping up against the coast of Australia. It becomes feasible. You can conceptualise how that voyage, that journey might have happened.
Today, this would be a 350-mile journey. But in prehistoric times, the coastlines were very different. "60,000 years ago, the planet was in an ice age. As great ice sheets were spreading across the northern hemisphere, so much of the world's water was frozen. Sea levels had fallen by 400 feet. The gap between Southeast Asia and Australia had shrunk. When people made the crossing, the two continents may have been as little as 60 miles apart. No one knows how many people first arrived in Australia. It may have been dozens, hundreds, perhaps 1,000. But according to the genetics, there was just a single wave of migration, which then stopped.
It seems like there's one entrance to Australia, and then people are, so to speak, spreading across the continent, occupying the continent. And this, as far as we can see, that only happens once.
Around 55,000 years ago, humans began exploring Australia. 40,000 years before they reached North America. Here they were truly alone, cut off from the rest of the world, in a virgin continent.
But these early Africans did not just stop at populating Australia, Melanesia, and Southeast Asia. They would also go on to populate the Americas as well, over 50,000 years ago. These African migrations into the Americas far predating the much later Mongoloid migrations into the Americas around 13,000 years ago. These Africans were living in the Americas before Mongoloid people even existed. Just as huge landmasses connected Australia and Papua New Guinea and many of the islands of South Asia and Australo-Melanesia during the Paleolithic, a land bridge connected Asia to the Americas. This land bridge was known as Beringia. These first African pioneers would cross Beringia and enter into these new untouched virgin lands, becoming the first Americans. These early Americans who migrated from Africa are best described as Paleo-African Americans. And like Omo I or Kafsa or many modern or ancient Australo-Melanesians or modern or ancient Sub-Saharan Africans, these early Paleo-Americans had the same ancestral African or Negroid phenotype. Thus, all early skulls in the Americas are overwhelmingly African. This has been well studied and well documented for decades, many having a very clearly Negroid or Africoid morphology. The Africanity of these early skulls and ancient remains serving as unequivocal evidence for the early populating of the Americas by African peoples over 50,000 years ago and during the initial out-of-Africa migration. This is Luzia, dated to 11,500 years ago, and like all Paleo-Indians, she has that distinctive Negroid or Africoid phenotype. Her remains were found in a cave in Brazil, where we happen to find large amounts of these early Negroid or African Paleo-Indians. This is Apiuna. This gentleman's remains are over 10,000 years old, found in Lagoa Santa, Brazil, just like Luzia. And like Luzia, he has that typical African or Negroid phenotype, which was common amongst the first Americans. To quote Nevis, in this study, we compare the cranial morphology of several late Paleo-Indian skeletons uncovered at Santana de Riacho, central Brazil, with worldwide human cranial variation. Malinobis distance and principal component analysis are used to explore the extracontinental morphological affinities with the Brazilian Paleo-Indian sample. Santana de Riacho is a late Paleo-Indian burial site where approximately 40 individuals were recovered in varying states of preservation. The site is located at Lagoa Santa. The first human activities in this rock shelter date back to the terminal Pleistocene, but the burials are bracketed between circa 8,200 years ago and 9,500 years ago. The collection contains only 6 skulls well enough preserved to be measured. The Santana de Riacho late Paleo-Indians present a cranial morphology characterized by long and narrow neurocrania, long and narrow faces with low nasal apertures and orbits. The multivariate analysis showed that they exhibit strong morphological affinities with present-day Australians and Africans, showing no resemblance to recent Northern Asians and Native Americans. These findings confirm our long-held opinion that the settlement of the Americas was more complicated in terms of biological input than has been widely assumed. The working hypothesis is that 2 very distinct populations entered the New World by the end of the Pleistocene, and that the transition between the cranial morphology of the Paleo-Indians and the morphology of later Native Americans, which occurred around 8,000 to 9,000 years ago, was abrupt. This, in our opinion, is a more parsimonious explanation for the diversity detected than a long local microevolutionary process mediated by selection and drift. The similarities of the first South Americans with sub-Saharan Africans may result from the fact that the non-Mongoloid Southeast Asian ancestral population came ultimately from Africa, with no major modification in the original cranial plan of the first modern humans. It is recognized that the human population that arrived in Australia around 50,000 years ago was the product of an expansive movement out of Africa. Following the tropical areas of southern Asia, this route of expansion represents one of the first offshoots of modern humans out of Africa. This is why Australians and Africans still form a superpopulation unit of morphological affinity. Therefore, the similarities of the first South Americans with Australians are easily explained if we accept that both populations shared a common ancestral population in mainland East Asia, most probably Southeast Asia. A population expansion from Asia to the New World before the Mongoloid traits were fully developed in the Old World was actually predicted more than 50 years ago by Birdsell in 1951. The similarities of the first South Americans with sub-Saharan Africans may result from the fact that the non-Mongoloid Southeast Asian ancestral population came ultimately from Africa, with no major modification in the original cranial plan of the first moderns. Nevis et al. Have already suggested a possible historical connection among early modern humans, Skull and Kafsa in Israel, UC 101, Paleo-Indians, and recent Africans and Australians, based on their cranial morphology. These early African migrants peopling the Americas tens of thousands of years before the much later Mongoloid migrations from Siberia and Northeast Asia, around 13,000 years ago.
All native peoples in South and North America belong to a racial type known as Mongoloid. They're descended from the ancient peoples of Siberia. During the Ice Age, 12,000 years ago, there was a land bridge between Siberia and Alaska. Mongoloid peoples were the first to enter the New World, or so experts thought. The Brazilian finds point to a very different story of the discovery of the Americas. The paintings on these rock shelters are much older than the Indians. Here, hunters chased giant armadillos, an animal that flourished during the Ice Age long before the arrival of the Indians. There's a playful feel to many of the scenes. Here, a group of men are standing on each other's shoulders, just like a circus act. And here, a rare romantic moment. The scenes conjure up a lost world of innocence. How long ago was this primeval paradise? French archaeologists have just finished digging in Pedra Furada, the largest of the rock shelters. Their aim was to go back in time to a period well before the arrival of American Indians. Every foot they dug took them thousands of years back into prehistory. When they reached layers 40,000 years old, they found these. pieces of quartzite that look remarkably like stone tools. The edges on one side of the stones were flaked off. Were these stones shaped into cutting tools by human hands? If so, then the history of the discovery of the Americas would have to be rewritten. But if people had been here before the American Indians, surely there would be other traces of their presence. The archaeologists continued to dig, down to depths 50,000 years old. And then they found this. Fragments of animal bone and charcoal. For Anne-Marie Passy, one of the archaeologists involved in the dig, these fragments were proof of human occupation.
We found structures shaped like hearths. Next to the charcoal, depending on the period, we sometimes found food leftovers of animals they may have eaten.
The Brazilian finds show that the New World was discovered tens of thousands of years earlier than previously believed, certainly well before the time of the American Indians. But who were these pioneers? A much earlier wave of Mongoloids, or another race altogether? Clues to the identity of the first Americans are emerging in rock shelters in the northeast and southeast of Brazil. Archaeologists there have recently unearthed human remains. Prehistoric skulls were found buried in layers of soil 9,000 to 12,000 years old. They are the oldest skulls in the Americas. And this is the oldest of them all. The skull of a young woman nicknamed Luthia by scientists. Can she tell us who the first Americans were? Walter Neves is a physical anthropologist at Sao Paulo University in Brazil. He has been using a standard and reliable archaeological measure, the shape of the skull, to find out what race she belonged to.
91.
He fully expected Luthia to be a Mongoloid, an ancestor of the American Indians.
91.
But then he fed the measurements into the computer.
When we start running the computer and seeing the results, it was amazing because we realized that the statistics, the quantitative analysis we were doing was not showing these people to be Mongoloid. In fact, the analysis was showing these people was anything except Mongoloid.
So who was Luthia? And where did she come from? To find out, the skull was taken to a hospital in Rio de Janeiro to begin the process of reconstructing her face. The first stage was to make a three-dimensional CAT scan of Luthia's skull in order to build a replica. The cast was then given to Richard Neve of the University of Manchester in England, one of the world's leading forensic artists, to recreate her features.
That, to me, is a Negroid face that has all the features that you associate with a Negroid face. The proportions of the face, it doesn't say anything about it being Mongoloid.
Luthia belongs to a race found historically along the rim of the Indian Ocean, in East Africa, in the islands of South Asia, and in Australia and Melanesia. Was this then the face of a first American? Her reconstruction is confirmed by measurements Walter Nevis has taken of all his skulls.
The first reaction was not to believe in it. But as the results, repeated so many times, and the result is exactly the same thing. They are very similar to nowadays Aborigines and Africans, and no similarity at all with Mongoloids in Asia or with American Indians.
But how could Luthia be African and Australian? According to Walter Nevis, there's a very simple explanation. Luthia is a bit of both. The first humans originated in Africa. It's known that around 100,000 years ago, waves of people migrated out of Africa. One such wave went east, and 60,000 years ago, it reached Australia. Luthia, it seems, belonged to a race of humans descended from Africans, a race that eventually became today's Australian Aborigines. At the southernmost end of the continent lies Tierra del Fuego, a group of islands isolated from the mainland by the Strait of Magellan. The oldest skull found in this part of the world is 9,000 years old. It too was measured by Walter Nevis.
This skull also shows a strong similarity with Australians and no similarity at all with nowadays Indians or Mongoloids. Okay, so even in the extreme southern cone of the Americas, okay, was inhabited by these people that has nothing to do with Mongoloids.
Thus, these early Africans that first populated Australia and Melanesia would also go on to populate the Americas as well, bringing with them that ancestral African phenotype. To quote Nevis, increasing skeletal evidence from the USA, Mexico, Colombia, and Brazil strongly suggests that the first settlers in the Americas had a cranial morphology distinct from that displayed by most late and modern Native Americans. The Paleo-American morphological pattern is more generalized and can be seen today among Africans, Australians, and Melanesians. Here we present the result of a comparative morphological assessment of a late Paleo-Indian early archaic specimen from Capilina Burial 2, southern Brazil. The Capilina skull was compared with samples of 4 Paleo-Indian groups from South and Central America and worldwide modern groups from W. W. Howell's studies. In both analyses performed, classical morphometrics and geometric morphometrics, the results show a clear association between Capilina Burial 2 and the Paleo-Indians, as well as Australians, Melanesians, and Africans, confirming its Paleo-American status. The increasing evidence that all late Pleistocene and early Holocene human groups from South America are characteristically non-Mongoloid has major implications for the colonization of the Americas, as argued by one of us since the end of the 1980s. Even if few studies with large samples from single sites have been carried out so far with Paleo-Indians, it is evident by now that South America, Central America, and possibly North America were populated by human groups with a more generalized cranial morphology before the arrival of the Mongoloids. Since this more generalized morphology, Australo-Melanesian-like, was also present in East Asia at the end of the Pleistocene, transoceanic migrations are not necessary to explain our findings. As presented in detail elsewhere, the arrival of an Australo-Melanesian-like population in the Americas is easily accommodated under what is presently known about the place of origin and the routes taken by modern humans in their first long-distance dispersions. Accordingly, a population that began to expand from Africa around 70,000 years ago reached Southeast Asia by the middle of the late Pleistocene, carrying with it a cranial morphology characterized by long, narrow neurocrania and narrow projecting faces. We postulate that after reaching Southeast Asia, this stem population gave rise to at least two different dispersions. One took a southward direction and arrived at Australia around 50,000 years ago. Sometime between 50,000 and 20,000 years ago, a second branch dispersed towards the north and arrived in the Americas by the end of the Pleistocene, bringing with it the same cranial morphology that characterized the first modern humans. When the classical Mongoloid cranial morphology appeared in northeastern Asia, either as a local response to extreme environmental conditions or as the product of a migration from northern Europe, a new expansion of northern Asians reached the New World, bringing with it a cranial morphology characterized by short, wide neurocrania and broad, retracted faces. These observations of early African migration into the Americas has also been further validated by ancient African genetics found throughout groups of Amerindians in Southern America, specifically in Brazil. These groups having ancient African ancestry, linking them to the founding populations of the Australian Aborigines, Andaman Islanders, and Papua New Guineans. To quote Skoklund, genetic studies have consistently indicated a single common origin of Native American groups from Central and South America. However, some morphological studies have suggested a more complex picture whereby the Northeast Asian affinities of present-day Native Americans contrast with a distinctive morphology seen in some of the earliest American skeletons, which share traits with present-day Australians, indigenous groups in Australia, Melanesia, and islands of Southeast Asia. Here we analyze genome-wide data to show that some Amazonian Native Americans descend partly from a Native American founding population that carried ancestry more closely related to indigenous Australians, New Guineans, and Andaman Islanders than to any present-day Eurasians or Native Americans. We have shown that a population Y that had ancestry from a lineage more closely related to present-day Australo-Melanesians than to present-day East Asians and Siberians likely contributed to the DNA of Native Americans from Amazonia and the central Brazilian plateau. This discovery is striking in light of interpretations of the morphology of some early Native American skeletons, which some authors have suggested have affinities to Australo-Melanesian groups. The largest number of skeletons that have been described as having this craniofacial morphology, and that date to younger than 10,000 years ago, have been found in Brazil, home of the Surui, Caritiana, and Zavante groups, who show the strongest affinity to Australo-Melanesians in genetic data. An open question is when and how population Y ancestry reached South America. There are several archaeological sites in the Americas that are contemporary to or earlier than Clovis sites. The fact that one individual from a Clovis context who has yielded ancient DNA had entirely first American ancestry suggests the possibility that population Y ancestry may be found in non-Clovis sites. Regardless of the archaeological associations, our results suggest that the genetic ancestry of Native Americans from Central and South America cannot be due to a single pulse of migration south of the late Pleistocene ice sheets from a homogeneous source population, and instead must reflect at least two streams of migration, or alternatively a long drawn-out period of gene flow from a structured Beringian or Northeast Asian source. The arrival of population-wide ancestry in the Americas must in any scenario have been ancient. While population-Y shows a distant genetic affinity to Andamanese, Australian, and New Guinean populations, it is not particularly closely related to any of them, suggesting that the source of population Y in Eurasia no longer exists. Furthermore, we detect no long-range admixture linkage disequilibrium in Amazonians as would be expected if the population Y migration had occurred within the last few thousand years. It should be understood when Skoklund says, while population Y shows a distant genetic affinity to Andamanese, Australians, and New Guinean populations, it is not particularly closely related to any of them, suggesting that the source of population Y in Eurasia no longer exists. This is because Papua New Guineans or Andaman Islanders or Australian Aborigines didn't travel to the Americas to populate it. The signal they're detecting is African genetics. The signal is of the African ancestors of the Papuans, Onge, and Australian Aborigines. It's the original African populations that first populated Australia, Melanesia, and the Andaman Islands, and of course the Americas as well. This population Y were the original African migrants of South Asia and Australo-Melanesia. All of these places, whether it be Australo-Melanesia or the Americas, were first populated by Africans during that initial out-of-Africa migration over 50,000 years ago. Another piece of genetic evidence that incontrovertibly connects Africa to the Americas is the presence of HTLV1, also known as human T-cell lymphotropic virus 1. This virus and its genetically inherited SNPs, or antigen alleles, namely HLA-A26 and HLA-A36, both originate in Western Central Africa. This disease has a particular transmission methodology. It can only be transferred through cell-to-cell transmission, usually being passed primarily from mother to child through breast milk or from male to female through semen and sexual intercourse. As stated before, this virus originates in Western Central Africa, where its greatest diversity and oldest strains can be found, as well as the hereditary antigen alleles associated with it. As we can see, all HLA alleles, which now have a worldwide distribution, can be found in Western Central Africa, where they first originated over 75,000 years ago. Today, HTLV1 has a worldwide distribution, but its most endemic areas are where human beings have lived the longest, namely Western Central Africa, where the virus originates, and South Asia and Austromelanesia, as well as the Americas, some of the first places populated by peoples out of Africa, being found in high frequency amongst the indigenous Australian Aborigines, Melanesians, and Japanese. Japan having the highest frequency of HTLV1 outside of Africa. This may be due to the fact that the Japanese are descended from ancient Western Central Africans bearing Y chromosome haplogroup D. And lastly, it is found in very high frequency in the Americas, namely in Brazil and South America. The indigenous tribes of Brazil also have a very rare and particular strain of human T-cell lymphotropic virus known as HTLV2. This version of HTLV has its origin amongst the pygmies of Western Central Africa. This transcontinental diffusion of HTLV1 and HTLV2 not only brought these African viruses into the Americas, but their respective antigen alleles as well. To quote Sonoda, the serological surveillance revealed transcontinental dispersal of HTLV1 in the prehistoric era that started out of Africa, spread to Austromelanesia and the Asian continent, then moved to North America, and through to the southern edge of South America. This was highlighted by an Andean mummy study that proved ancient migration of Paleo-Mongoloid HTLV1 from Asia to South America. transcontinental dispersal of HTLV-1 with human migration. Human T-cell lymphotropic virus type 1 is transmissible from mother to infants through breast milk, from male to female through semen, and to blood recipients through HTLV-1 carriers' lymphocytes. All the transmission routes effectively localize HTLV-1 infection foci within particular family ethnic groups. Efficacy of vertical transmission of HTLV-1 was confirmed by recent studies of HLA concordance between mothers and infants, likely mediated by either immunological or virological synapse of HTLV-1 infected T cells. Thus, serological and virological surveillance allowed location of HTLV-1 positive ethnic groups across the world, as depicted in Figure 1. By virtue of the genomic stability of HTLV-1, we could trace the transcontinental dispersal by phylogenetic relationships of viral isolates from various ethnic groups. The early transmission of HTLV-1 to South America was highlighted by the discovery of ancient HTLV-1 DNA preserved within an Andean mummy. The mummy was buried 1,500 years ago in the desert of Atacama, northern Chile, and the body was kept intact in the dry climate with salty and alkaline soil. When bone marrow samples of mummy femur were processed to extract DNA by a glass meal method, followed by PCR amplification of HTLV-1 DNA, nucleotide sequences of the mummy's HTLV-1 tags and LTR DNA were very similar to HTLV-1 isolated from the contemporary Atacama tribe and the nearest neighbor of Chilean indigenous people. Of particular interest, they were closely related to the HTLV-1 of Ainu, southern Japanese. The transcontinental migration of HTLV-1 was supported by studies showing two lineages of HLA-A alleles in association with HTLV-1 endemic populations in the world, one being HLA-A26 and the other HLA-A36, and both originally evolving in African blacks and then becoming dispersed to Melanesians, Israeli Jews, Asian Indians, Southwestern Japanese, Sakharan natives, and North and South Amerindians, and today carried by Caribbean South American black peoples. It is likely that ancient HTLV-1 passing peoples in Africa moved to the Asian continent, then passed the Bering Strait to North America, and further to the southern edge of South America, as depicted in Figure 5. Thus, both the anthropological evidence showing the clear African or Negroid morphology of early Paleo-Americans, as well as the genetic evidence connecting the Americas with the early African populations that would become the Australians, Melanesians, and other South Asian groups, as well as the endemic nature of not only HTLV-1, but also the much more rare HTLV-2, as well as their ancestral African antigen alleles, all overwhelmingly support the idea of the early peopling of the Americas by populations of Africans during the initial out-of-Africa migration. To quote Kanzaki, based on recent serological detection, isolation, and characterization of HTLV-3 and HTLV-4 among African populations in Central Africa, and additional unrefutable evidence of early human migration from Africa and Australia to the American continent previous to Asiatic population migration lead us to hypothesize that human descendants of mixed Americans and Africans or remaining Africans explain the very frequent presence of Western blot indeterminate results for HTLV-1 and 2 that we and other groups have been detecting, and also the unusual absence of HTLV-2 infection among some relatively homogeneous ethnic native human populations in the American continent. According to different authors, during the Pleistocene-Holocene transition period, at least two different human populations migrated to the American continent. Cranial anatomic analysis recognized two peculiar human morphologic patterns related to Asiatics and Africans. So, contrary to the common sense, one of these groups, the Asiatics, were not the first human groups to migrate to the American continent, but the African group, probably coming from Oceania and or Africa, Confirming these findings, Nevis et al. Emphasizes that the colonization of the New World occurred by two distinct succeeding biological populations, an early one with a cranial morphology similar to that found today in the African and Australian continents, and a later one with a morphology similar to that found today among northeastern Asians. Nevis et al. Also have been finding increasing skeletal evidence from USA, Mexico, Colombia, and Brazil, strongly suggesting that the first settlers in the Americas had a cranial morphology distinct from that displayed by most late and modern Native Americans. Based on these research results and our unusual findings of HTLV-1 infection among Amazonian Amerindians and indeterminate Western blot profiles, we hypothesized that early human populations, the Austromelanesians and Africans, colonized the American continent 12,000 to 35,000 years in the past, previous to the migration of Asian populations. So not only do genetics support the early peopling of the Americas by Africans during the initial out-of-Africa migration, but the anthropological record as well, all Paleo-Indians possessing that distinctive African or Negroid skeletal morphology. This early populating of the Americas is also supported by the actual archaeological record of the Americas. For a long period of time, it was suggested that people only arrived in the Americas about 14,000 years ago. These were the Clovis people, and these were Mongoloid people from northern Asia, who entered into America at the end of the Ice Age and the last glacial maximum. This limited view of American history has been thoroughly destroyed, the Clovis first model being completely destroyed over the last several years, as several finds have refuted the idea of Clovis first. Several sites have been found throughout the Americas dating to 30,000 years ago, 50,000 years ago, 100,000 years ago. And it should be understood that most sites that were dated before Clovis were always met with intense and often acrimonious scrutiny and criticism from many North American archaeologists. This is largely due to the African or Negroid skeletal morphology of all early Paleo-American remains. Of course, the idea of Africans being the first people in the Americas is off-putting to a lot of people with racial biases and in the field of archaeology, even if they're supposed to be impartial and non-biased professionals. The idea of race and the Africanity of the first peoples of the Americas is the unspoken elephant in the room in modern American archaeology. Prior to the last several years, anyone that suggested a site was older than Clovis in the Americas was met with harsh, illogical, and acrimonious arguments. All of this to try to maintain that Clovis first model. But it should be noted that South American archaeologists and even many European archaeologists have historically had none of this racial bias, and for decades have had no problem whatsoever seeing that the first people in the Americas were Africans. They've in fact been saying this for decades, simply being honest and just following the archaeology and the evidence, being genuine and professional scholars. One of the oldest sites showing very clear and early evidence of people living in the Americas far before Clovis is the site of Serra de Capivara, of course found in Brazil. These paintings dated back to over 30,000 years, while occupation at this site and nearby sites go back as far as 100,000 years.
Man was here 100,000 years ago. We think that probably they came from Africa. The currents and the wind came from Africa till here.
This is Simon Romero for the New York Times in the Serra da Capivara National Park. Here, in the forbidding thorn forests of Brazil's Piauí state, archaeologists say their discoveries are contributing to a pivotal reevaluation of how humans first came to the Americas.
What archaeologists are finding here at this incredibly remote site
is evidence of human habitation going back tens of thousands of years, which totally upends previous theories of human migration across the Bering Strait, across North America, across the Amazon rainforest, and into this part of Brazil.
Today we have 945 sites with rock art.
French-Brazilian archaeologist Dr. Nied Guidon has overseen digs here since the 70s. New findings throughout the hemisphere are upending the Clovis first theory, a prevailing belief of 20th century archaeology in the United States that humans began arriving in the Americas about 13,000 years ago. The research in Brazil is trying to determine how early lands empty of humankind were settled.
They have these structures to make fire. We took samples of the stones that were heated by the fire, and it was possible to date by a thermoluminescence to 100,000 years.
A beringia, só como ponte biogeográfica para se atravessar, para que os grupos humanos atravessassem da Ásia para a América, provavelmente não foi a única ponte nem num único período.
Dr. Guidon's assertion that humans lived here 100,000 years ago has not been widely embraced, but research up and down the Americas increasingly reveals signs of human habitation long before Clovis hunters began appearing. Dr. Guidon first came to Piauí to study the rock art left by ancient people. Excavating beneath these striking paintings, she says she found stone tools and remnants of ancient fires. Archaeologists have also unearthed more recent burial sites and fossils of extinct megafauna like giant sloths and saber-toothed cats. Critics dispute some of their findings, claiming that sharpened stones could have been made as the result of rockfall or by monkeys. But the researchers here say the stones show clear evidence of human craftsmanship.
Peças que teve a sua quebra pelo que a gente chama de aquecimento, ela vai ficar bem côncava e bem convexa. A outra parte sem orientação de retirada. Quando antrópica, não, quase sempre você vai conseguir orientar exatamente de onde foi feita a retirada.
Dr. Guidon is now retired from active research, but teams of Brazilian and European archaeologists continue the work she began at a world-class institute and museum she founded here. In 2013, a French-led team published findings placing the human presence in Piauí as far back as 22,000 years. As scholars advance beyond Clovis first, this region, long mired in poverty, is now emerging as a flashpoint for research and discovery.
One of the oldest sites showing a clear presence of peoples in the Americas at a very early date is the Suruti Mastodon site, dated to over 100,000 years ago. And this is not too surprising. As we know, the peoples of Papua New Guinea in part descend from a much earlier wave of modern humans out of Africa, dating back as much as 120,000 years ago.
In November 1992, paleontologists from the San Diego Natural History Museum were conducting routine monitoring of a construction site on State Route 54 here in San Diego, when field paleontologist Richard Suruti spotted several fossil bones and portions of a tusk. It took 22 years to piece the story of these fossils together, and the picture it reveals just might change everything. As Suruti explored the site with Dr. Tom Demaray, they found fragments of distinctive molars showing that they had discovered the bones of a mastodon. But something was puzzling. Many of the bones were strangely fractured or missing entirely, and there were several large stones that seemed out of place in the surrounding fine-grained sediments. To Richard, this was starting to look not like a paleontological site or a fossil site, but instead like an archaeological site, like the preserved evidence of human activity. As work on the site continued, questions about its age remained unanswered. At the time, the oldest evidence of humans living in North America came from sites that are around 14,000 years old. But mounting evidence suggested this site was much older, possibly dating to long before any humans were thought to have arrived on the continent. The first step to solving this riddle was to confirm that the site was, in fact, an archaeological site. Dr. Steve Holen had the expertise to help. Using the femur bones of recently deceased elephants, Holen had conducted 2 separate experiments in which he demonstrated that a stone used as a hammer causes fresh limb bones to fracture in a distinctive way. No known geological process or animal scavenging produces the same fractures in bones. In 2008, when Holen first traveled to San Diego to study these specimens, he confirmed the same breakage patterns. Furthermore, the San Diego site had 5 large rocks that could have been used to produce the breakage. The broken bones were concentrated in 2 separate clusters, each around 1 rock. The other large rocks lay nearby. Could these have been used as tools? To help answer this question, the team consulted Dr. Richard Fulliger, an expert in use wear features on stone tools. Richard compared the impact and wear marks on the stones found at the San Diego site with other stone tools. Sure enough, the large stones showed the same abrasions, fractures, and scars that only occur on stones used as hammerstones and anvils. No known geological process produces the same patterns of wear. A clear picture was starting to emerge of 2 distinct areas where early humans were using rocks to break bones and teeth. But couldn't a flood or other event have rearranged the rocks and bones? Holen, who has studied sites where this happened, says no. At other sites, gravity sorted bones by density and size. This wasn't the case here, where everything from small molar fragments to large rocks were distributed, unsorted, around the site. The researchers were unanimous. They had uncovered an archaeological site containing portions of an extinct mastodon. But a critical question still remained unanswered. How old was the site? In 2011, almost 2 decades after the mastodon's discovery, advances in technology provided the answer the team was looking for. Tom Demarais sent several of the mastodon bones to Jim Pacey's, who used state-of-the-art radiometric dating methods to determine their age. The mastodon bones, which were still fresh when broken with stone tools, were around 130,000 years old. What does this mean? It means that early humans were living in North America at least 115,000 years earlier than previously thought. This site's discovery reframes our understanding of when humans first arrived on this continent. It also raises as many questions as it answers. Who were the humans at work at this site, and how did they reach North America? Were they an early, failed attempt to colonize a new part of the world? Where are the other sites that show human activity in North America that are as old as this one? Where else should scientists look for evidence? Asking questions is at the heart of scientific discovery. Science is always seeking the best explanation that fits the evidence when new information is uncovered, no matter what we thought was true before. Fossils tell stories. This is not the end of this story, but simply the beginning.
As I said, sites like these have recently destroyed the much-espoused Clovis first model, which posited that Mongoloid peoples from North Asia were the first in the Americas. This model being completely destroyed, as new sites showing much older occupation are being found every year. Not surprisingly, one of the most recent finds is in Santa Helena in central Brazil, home of Apayuna and Luzia, and countless other Negroid or African Paleo-Americans. This site dates back to 27,000 years ago. Here we find the oldest jewelry in the Americas, 3 finely polished and sculpted osteoderms of a giant ground sloth. These beautifully shaped pendants would have been worn around the neck as jewelry by these early Paleo-Indians, almost 30,000 years ago. These finely shaped pieces of jewelry being the oldest jewelry ever found in the Americas. Multiple multidisciplinary tests were run on these artifacts to verify that these osteoderms were indeed created and modified by humans and not the result of fleas, rats, or other natural processes. These pieces of bone showing clear signs of intentional drilling and sculpting. And not surprisingly, even more evidence of early man has been found in South America. These butchered remains of megafauna go back over 21,000 years and are found in the Pompeian region of Argentina.
Fossil bones found in Argentina with cut marks suggestive of butchering indicate humans were present in southern South America some 21,000 years ago, according to researchers. That's earlier than previously thought. The bones were from a large armadillo relative unearthed in 2015 in the Buenos Aires metropolitan area. A study on the finding was published on Wednesday. Here's anthropologist and study lead author Mariano del Papa.
Las marcas que encontramos, en principio, son
The marks we found are linear, with no more specifications. A detailed analysis led us to determine these were marks of cuts, and the only ones who could make them are human beings. They are not marks of stamping from other animals, not rodents or bites of carnivores. Another evidence we found is that these bones had not been disjoined, as they preserved their natural joints.
The researchers said the cut marks on the bone appear to have been made by people using stone tools, though no human fossils were found at the site. Cut marks were found on the pelvis, tail, and body armor of the specimen. The timeline for the peopling of the Americas has been a matter of debate. The new findings represent the oldest evidence of both the presence of Homo sapiens and human interaction with large animals in southern South America during the height of the last ice age, researchers said.
But perhaps the most landmark site found to date is the site of White Sands, New Mexico, which possesses the fossilized footprints of human beings and megafauna living side by side from over 23,000 years ago. This site drastically pushed back the presence of humans in the Americas, giving incontrovertible proof of the early peopling of the Americas during the initial out-of-Africa migration and before the onset of the last glacial maximum. This site serving as the final nail in the coffin for the Clovis first model.
The vast and dry gypsum landscape in what is now New Mexico was once a giant lake. Archaeologists began digging through the layers of sediment laid down over tens of thousands of years, and then, clear as day, they came upon this: fossilized human footprints. Carbon dating of ancient seeds found among the footprints suggested a time long before it was thought possible. More testing of more layers confirmed the footprints were children walking by an ancient lake almost 10,000 years before we thought humans had arrived in North America.
Radiocarbon dating with a very high degree of certainty, and we were astounded at just how old they turned out to be.
In Africa, skulls of Homo sapiens date earlier than 100,000 years ago. From there, great migrations began, first through the Middle East to South Asia, and later to Australia and to Europe, 40 to 50,000 years ago. More recently, humans crossed over what was then a land bridge that existed between Siberia and North America. It was believed they came only after the glaciers of the last ice age had begun to recede around 13,000 years ago. The new footprint suggests humans came before the ice-blocked migration and came to the southwestern U.S. up to 23,000 years ago.
This discovery is pretty staggering because it says people were actually here before those routes closed up.
It's believed the early humans in North America would have coexisted with mastodons and other primitive species. They may well have been the forebears of the indigenous people in the Americas. And the study suggests an early society existed here for at least 2,000 years.
You can just kind of picture the kids. It's like, oh, let's go down, play by the lake. Or their parents say, go get some water. It's just like step, 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 step. These little glimpses of activity and interaction over time are just fascinating to me.
Scientists say future research should focus on even older, undisturbed surfaces, where there may be earlier evidence of humans we didn't realize were already here.
And Clovis was recognized by the 1950s as the earliest well-recognized assemblage. There's also artifact assemblage that's called Western Stemmed in the Great Basin that are probably some as old as Clovis. But then this raised the question of immediately, of course, was there anybody here before Clovis? And that debate's been going on since the 1950s. And it's a endless debate, acrimonious debate at times. There are good reasons why this is that I won't get into. This debate persists. But it is one of the great debates in American archaeology. And also we're dealing with the last major human migration in human prehistory, the peopling of the Americas, much later, of course, than in Europe and elsewhere. Now, what we've got at White Sands, locality 2, as you have now seen from David and Matthew, unequivocal evidence of people walking around. For me, that has been a tremendous relief. We don't have to argue about broken stones or broken bones. Human footprints, unequivocal. There's no doubt, lots of them. It's astonishing to see this, on top of this astonishing record of megafauna. White Sands down here, the oldest in this group, and there are other sites, but I think these are probably the best cases in North America. This is particularly significant because the White Sands locality is the same age as the glaciers, the maximum extent of glaciers across North America. As most of you are probably aware, you've had these cycles of ice growth and decay, and 23 to 25,000 is when you had the last cycle of ice growth, and you had glaciers essentially from coast to coast covering all of Canada. This is a generalized map, but it shows you the ice coverage. There was an ice sheet over the Canadian Rockies, smaller one, and then a large one, and they would grow and merge and completely cover Canada for a while. And this is a map at probably right at the end of the age range for the end, as far as we know it, for the White Sands locality. And so that site is very clearly occupied by humans at the same time that the part of the continent is completely covered. So that raises a lot of questions about how did people get to New Mexico and when relative to this situation. In my last slide here, this is a textbook illustration showing the peopling of the Americas as the ice sheet shrank. And when Clovis arrived and the other groups arrived, showing various routes through the glaciers as they retreated or around the glaciers. But you can also work this the other way, because the glaciers grew out of these centers. So 100,000 years ago, 50,000 years ago, there was essentially no glacial ice covering Canada. But then as climate change, you started growing the glaciers and sea levels started going down. But there was probably some several tens of thousands of years when you could get from Asia into the Americas and get around or through the glaciers before the last glacial maximum. So this is an argument that we've put out that possibly we're seeing a population that had been in place for some time, which makes the whole issue of the peopling of the New World, pushes it back even farther. So, I mean, the site is so significant for so many reasons. And this is yet another one of the reasons why we're interested in it.
Sites like White Sands and Santa Elena and Serra de Capivara show clear and incontrovertible evidence of human occupation in the Americas far, far before Clovis. These various ancient sites predating even the mutations on that OCA2 gene responsible for light skin amongst East Asian peoples. These sites being occupied tens of thousands of years before the rapid expansion of Mongoloid peoples during the Holocene. In fact, at the time when people were living at White Sands, that typical Mongoloid phenotype had not even become widespread in East Asia yet, with several remains from China and Japan dating back some 20,000 to 12,000 years ago, still having that distinctive Negroid, Sub-Saharan, or Australo-Melanesian phenotype, that same ancestral phenotype from Africa. Ancient remains of these Negroid East Asians be found in sites such as the Zhoukoudian Cave near Beijing. As Professor Holliday stated, these early African migrants would have arrived in the Americas 50 to 100,000 years ago, before the closing of the ice corridor and the start of the Ice Age and the last glacial maximum.
But you can also work this the other way, because the glaciers grew out of these centers. So 100,000 years ago, 50,000 years ago, there was essentially no glacial ice covering Canada. But then as climate change, you started growing the glaciers and sea levels started going down. But there was probably some several tens of thousands of years when you could get from Asia into the Americas and get around or through the glaciers before the last glacial maximum. So this is an argument that we've put out, that possibly we're seeing a population that had been in place for some time, which makes the whole issue of the peopling of the New World, pushes it back even farther.
These early out-of-Africa populations would prosper in the Americas for millennia, until about 14,000 years ago. With the end of the last glacial maximum, we would see yet another migration into the Americas. These peoples would be the more Mongoloid, Northeast Asian populations we usually identify with Native American populations. Just as in South Asia and regions of Austro-Melanesia, we would see these two populations side by side. Today in these regions of Asia, we can still see these much older Negroid populations side by side with the more recent Mongoloid migrants who moved into these regions at the very end of the Neolithic. In the Americas, these Mongoloid and Negroid populations would mix, and throughout the Americas, we can find both of these distinct skeletal morphologies side by side in certain sites. But it should be known, these Paleolithic African migrations were not the last migration of Africans into the American continent. And in fact, some of these Mongoloid populations would in fact have African populations amongst them. As we know, during the Neolithic, the Sahara, which today is a barren desert, was lush and green, which led to massive movements out of Africa into new regions of Eurasia. These Africans would bring pottery, agriculture, and urbanism to several new regions of Eurasia. But they wouldn't stop there. They would also migrate deeper into East Asia, as well as into the Americas. As we know, throughout Europe, the Levant, and Asia, it is common to find the remains of ancient African individuals, many of them bearing the ancestral African Y chromosomes, such as A, B, BT, CT, and various E lineages, as well as possessing those distinctive Africoid or Negroid features. The remains of these ancient Africans found everywhere from Spain to India. So, just as we find multiple remains of Africans throughout these regions, we also find them in the Americas as well. This ancient African connection is most likely the cause of many of the shared cultural traits that are found between the Americas and Africa. Of course, the most striking is the use of pyramids, as well as the intentional use of mummification. The oldest mummy in Africa is a small black boy found in the Green Sahara, dated to over 6,000 years ago, 1,000 years before the first mummies in Egypt. And of course, we find mummies in the Americas that are thousands and thousands of years old. The oldest mummies of the Americas were of the culture of the Chinchorro of southern Peru and Chile. Some of these mummies are 7,000 years old. The Chinchorro people were expert fishermen, their lives revolving around the lakes and rivers of Southern America. Not surprisingly, the first stone-building civilization is also in South America, not far from the Chinchorro. These were the people of Norte Chico. The people of Norte Chico built the first urban centers in the Americas, as well as the first step pyramids. These were a completely peaceful people, with no indication of warfare and ritual use of psychedelics. So, just as we find the remains of ancient Africans throughout the Mediterranean, the Levant, and India, we also find the remains of these ancient Africans in the Americas. Dated to between 3972 BC and 3806 BC, the remains of this ancient mummified African, not surprisingly, can be found amongst the Chinchorro, the oldest mummifiers of the Americas, in their ancestral heartland of Arica in Chile. This ancient African mummified individual bore Y haplogroup E1B1A1 A1C2C3C. This E1B1 lineage, of course, originates in Western Africa and is found commonly amongst Niger-Congo peoples of the African continent. We also find African individuals dated to between 4310 BC and 3940 BC. This ancient African individual residing in Neolithic Canada. This individual carried the ancestral African Y haplogroup A, now found in the dense tropical regions of the African continent. This deep-rooted African Y chromosome being synonymous with sub-Saharan Africa. We also find another pre-Columbian mummy, dated to over 1,000 years ago. This mummy was found near Chihuahua, Mexico, and carried the African Y chromosome CT. And not surprisingly, we also find several other pre-Columbian individuals bearing these African A, CT, BT, and E lineages throughout the Americas, and even in other regions of Asia, speaking to the large spread of African migrants during the age of the Green Sahara and the Neolithic Revolution. So where did all these black people go? Where did all these Africans go to? Where are the Paleo-Americans that first migrated here from Africa during the Paleolithic? Where are the African migrants that came here during the Neolithic? If we have all these genetics and skulls and skeletal remains, where did all these black people go to? They're here. I'm one of them. A lot of African Americans are descended from these ancient African groups. For example, this is my Ancestry.com, and of course I'm black, but I'm also 5% Native American. This is on my mother and father's side. This is me and my sister's friend Julie, and me and my sister share a birthday, so this is Julie mushing up my sister's face on our birthday. And Julie is Mexican, obviously. And her dad's Mexican. And he's a really cool cat, real funny guy. And he recently did a DNA test. And he was 40% African. 40%. Both West and East African. Which is typical for African Americans. Because, they're not usually just from one region of Africa. But Julie's dad doesn't look black at all. He's just a regular looking Mexican guy. But he has a very high percentage of African ancestry. That's more than certain Afro-Mexicans or even Afro-Latinos who look more, like me. So I'm saying all this just to illustrate how in the Americas, we're all mixed up. We're all mixed together. And a lot of this mixing happened before Europeans even arrived. We already lived on this continent together before the era of colonization. When the Americas were colonized, the first people that were enslaved were Native Americans. Over 5.5 million Native Americans were enslaved during the era of colonization. During that time, of course, people were brought over from Africa as well. If you were a Native American of African ancestry, you were just put with the other Africans and then labeled as colored or Negro. And this goes for North America, South America, Central America, the Caribbean. A lot of those people in those areas are descended from those Paleo-Americans that first migrated from Africa 50,000 years ago, 100,000 years ago, as well as African migrants that came here during the Neolithic. A lot of people are descended from those ancient populations. They just don't know it. Why is that? Because, unless you study pre-Columbian American archaeology, you've probably never heard of any of the stuff I've covered in this video. Who the heck knows about HTLV1 or, these genomes? I didn't even cover things like sickle cell. A lot of ancient Native Americans had sickle cell, which of course originates in Western Africa. So a lot of this stuff is not exactly hidden, but people don't usually talk about it. So, why would you know about it? But, thankfully with all of the research, all of the new archaeological finds, all of the new sites, you know, great archaeologists like Nevis, like Guidon, who have always said the first Americans were black people from Africa. They've been totally proven right over the last, you know, couple years. And eventually, you know, even if I were to do it myself, there will be documentaries about the first Americans, and they will be black people, you know, and that will mean a lot to, you know, a lot of kids in the Americas, whether they be in Caribbean or whether they be in Compton. You know, seeing the first Americans as black people will mean a lot to a lot of people. So, yeah, that's the end of that video. You know, thank you guys for watching. If you guys enjoyed the video, please, you know, drop a donation. These videos take a lot of work. But yeah, thank you guys for watching. See you next time. Later.